Hi student, in this video we are going to learn about the origin of cells, right? And we also will see how the theory about the origin of cell developed from time to time. Okay, so our journey will start at before 19th century, so a long time ago. Okay, now back then, the scientists believed that living things come from non-living things. Now in Latin, this kind of theory is called generatio spontanea, okay, which means that life could appear from non-living materials. And as you may have learned, that theory has to be supported by evidences. Now, so in the past, there was a scientist called Jean-Baptiste van Helmont. Perhaps that's how it pronounces his name. I'm not sure. Okay, so he lived in the 17th century. All right, and he did this experiment. Okay, this is called the recipe to create mice. So what you need, and he did this experiment to prove that, is dirty shirt plus wheat. So if you have dirty shirt and wheat, right, put them together and keep for 21 days. And after that, mice will appear from the combination or the mixture between dirty shirt and wheat. It is proven. It was proven. What is wrong with this? According to today's scientific method? Well, I'll leave it to you to think about, and we will discuss about it when we come back. Listen again. So back then, this experiment was accepted as, yes, the evidence for the generatio spontanea. But again, what is wrong according to today's scientific method? What is wrong with this experiment according to today's scientific method, right? That's for our discussion in the class. However, one day, a French biologist called Louis Pasteur, he discovered, he did an experiment that is so convincing, so it falsified the previous theory, generatio spontanea. This is what he did. Okay, so this is the equipment that he used. We call it like the swan neck flask. Okay, and this flask is filled with broth, like chicken broth and so on, which means that there's food in it, right? Comes from the meat or comes from the chicken meat and so on. All right. Now, what he did was this flask that is swan neck flask that is filled with the broth was sterilized. Okay, this is just to kill all microorganisms that might exist in the broth. All right, and after that, he kept the flask for days. And by doing this, air actually have access to go into the flask, but the microorganism will be trapped here because they are heavy, so no pressure that can lift them up and enter the flask. And he was right. The microorganism did not appear, which means that the broth remained clear for many days. All right. So what Pasteur wanted to show was that in generatio spontanea, the scientists of that theory who believe that theory, of course, they will say when broth mix with air, this will give birth to microorganisms. Now, according to this experiment, it is proven that after many days, there was no microorganism. Okay, like what I said in the previous slide, the broth in the swan neck flask remained clear or free from microorganism. Now, actually, it was not shown in the picture. He also did another set of experiment, the same swan neck flask, but this one was not boiled as the control. And of course, this flask that was not boiled, okay, must contain the microorganism and the broth. So the microorganism made the broth to be cloudy because, yeah, they they consume the broth, and they also produce waste that make the broth cloudy. Now, again, to prove or to make the conclusion even stronger, at the end of the experiment, Pasteur snapped the neck of the flask. And of course, by doing so, the microorganism now has an access to the broth inside the flask. And soon, the broth become cloudy. So the conclusion of the experiment is, look at this, even though the air can have an access, but not the organism, no organism can appear or alive in the broth. But when the air plus the organism, microorganism, enter the flask, then life is possible. So there is no generatio spontanea. It must be living things or microorganisms from the air outside that cause the microorganism to exist in the flask. So this is the conclusion from the Pasteur's experiment. Once again, I have to repeat that. Living things come from pre-existing living things, or now the theory is called biogenesis. Now, if we link to the cell theory, if we talk about this concept in the language of cell theory, I hope you still remember this point from the cell theory. All cells come from pre-existing cells, right? However, the next question is, if let's say the first cell, today's cells come from the pre-existing, and the pre-existing come from the pre-existing, and so on and so forth, we come to the first point. Where did the first cell come from? Oh no! Even Jackie Chan is so confused, all right? Where does it come from? Where did the first cell come from? The bad news is we don't know yet. But, well, let's be a bit optimistic yet, right? Hopefully, in the future, we will know. Okay, anyway, we don't have theory, we don't have any evidence, but we have hypothesis, right? Biologists made hypothesis that the ancient world, billion years ago, the world has what we call primordial soup, so it looks like this, okay? That might be supportive for the first simple organism to appear. So as you can see here, that the temperature is so high, yeah, it's very hot, right? And then there are meteorites and the electricity from the lightning and so on and so forth. So they thought like, wow, this harsh condition, this chaotic and so on, that make the molecules to collide, get energy, and so on and so forth, that might cause the first organism to appear. Now, there are two scientists, Stanley Miller and Harold Urey, that made an experiment that slightly supported this hypothesis. We will discuss their experiment in the next slide. So this is Stanley Miller and Harold Urey experiment. So this is their set of experiment. So I will explain the part of their experimental set. The first one is this. This is the gas chamber where there are also electrodes that spark electricity. So it's kind of like lightning stimulation. All right, and inside here are mixture of gas in primitive atmosphere, right? For example, like the ammonia, the carbon dioxide, and so on. Okay, now after that, the gas that has been electrified, they will enter to the condenser. Remember, they have ocean, so they can get cool, right? And then after that, they will become condensed and then heated again. Again, it's like the ocean. From here, they will be collected. And remember, back to the previous slide, the ocean, as you can see, it's boiling. It's boiling. It's really hot. So that's why it's heated. The water here, after this condensed, from the lightning, they will be condensed and then heated. And then again, of course, they evaporate and enter the gas chamber again. Electrified, cool down, collected, boiled up, and then electrified, and so on and so forth, again and again and again. Right. So again, this is the summary of the experiment. And the experimental set mimic the ancient world condition, which is the gases in the air here in this chamber, the lightning. Okay, I think I have to erase this a little bit. Yeah. Okay, so the gases in the air, which is here in the chamber, the lightning, which is here, the electrodes, and then the temperature heated up here, and then the water as the ocean here, right, in this bulb. All right. So after many cycles of heating, electrifying, condensing, boiling, they found amino acids. So they took some sample here, stop clocks for testing sample. So they take some of the liquid. Okay, they collect the liquid, and then guess what they found in the sample of the liquid? It's amino acid. Well, you will meet with amino acid soon in chapter 2, right? What is amino acid? Basically, amino acid, in short, I can say, that's part of a protein. And no non-living things that have protein, only living things have protein. So, well, indirectly, it's kind of support the hypothesis or create an evidence because protein is part of living things, but not yet a living thing. So that's why it remains as a hypothesis until today. But don't be sad, even though we don't have the theory for the origin of cell, at least we have the theory for the origin of the first eukaryote. All right, so let's listen to the history. So this theory was announced or declared by Lynn Margulis, and she was an American evolutionary theorist and biologist. And this is what she said. So let's first understand the meaning of symbiosis. Symbiosis happens when two living organisms living together. All right. Well, endo plus symbiosis means when larger cells takes in. Now, this is where the endo word comes from. Larger cell takes in a smaller cell by endocytosis, in a fascicle, but not digesting it. Well, unlike the endocytosis, like the video that we watched before, amoeba ingest a paramecium by endocytosis. In this case, the prokaryotes that is engulfed by endocytosis was not digested. They remain alive. Right? So for example, like mitochondria and chloroplast, Lynn said mitochondria happen when anaerobic bacteria, anaerobic means they don't need oxygen, they do not use oxygen, took in aerobic bacteria. So you can see from here. So this orange thing are mitochondria. So once there was a prokaryotes that they use oxygen for their metabolic processes. Well, this bigger cell do not. And this bigger cell do endocytosis makes the prokaryote that is aerobic to enter. And they become mitochondria. All right. How about chloroplast? Well, Lynn also said that chloroplast was created when non-photosynthetic bacteria, so this cell is non-photosynthetic, you cannot see any green color inside, took in photosynthetic bacteria. So this was once a prokaryote with a chlorophyll. So they can become a photosynthetic bacteria. And this bigger cell engulfed them and then become the chloroplast. And in addition to that, you also can see the reticulum endoplasmic. Lynn said that reticulum endoplasmic came from the plasma membrane that invaginate inside or folded inside. And then as you have learned in endocytosis, later on this plasma that is folded inside will disintegrate from the main plasma membrane and they become a separate organelle inside the cell. So maybe you ask me, but why is it a theory? Isn't it has to be supported by evidences? Yeah, don't worry. Let's talk about the supporting evidences. The first one, chloroplast and mitochondria are what? They have naked loop of DNA like prokaryotes. If you still remember, yeah, in the previous subchapter, the prokaryotes have naked DNA in the nucleoid region. And then also, the ribosome of prokaryote is 70S, while eukaryote, if you still remember, eukaryotes ribosome is 80S per. Okay, so while this chloroplast and mitochondria have 70S, so it's pretty similar to prokaryotes, isn't it? And they can create their own proteins. And also they have double membrane. If you still remember, when a cell do endocytosis, right? For example, like this, yeah, the endocytosis will make the whatever that is engulfed to be encapsulated by a fascicle. And you can see from this picture, the chloroplasts have double layer. So maybe it was once, all right, the fascicle that is used for endocytosis, while the inner membrane belongs to the prokaryote that was engulfed by the bigger prokaryote. All right. Now, in the next slide, you will see the picture of mitochondria with the DNA and also the ribosome. So I'll show you first here. Chloroplast DNA, see, this is a circular naked DNA similar to prokaryotes, and you also can see ribosomes here. All right, this is our ribosome. We will see the mitochondria after this. And here comes the mitochondria. I don't have to read it again because basically all these points are the same. Let me show you directly. Okay, these are the DNA. Again, these are naked DNA. Okay, and you can also see some ribosomes. All right, so those dots, okay, the blue dots here are the ribosomes. And the ribosome unit is not 80S, it's 70S. So it's so much not similar. It's basically the same like what prokaryotes have, 70S ribosome. Okay, so these three things that become supporting evidence. Both mitochondria and chloroplasts have naked loop of DNA like prokaryotes, 70S ribosome like prokaryote, and also double membrane as if they were taken by the process of endocytosis. And the last supporting evidence is both mitochondria and chloroplasts grow and divide like cells. So this is the picture that is produced by the electron microscope. You can see that actually this was one mitochondria, and at the middle part, I use another color, at the middle part it starts to divide. Okay, and slowly but sure this line getting bigger and then reach the end. All right, and when it reached the end, this one mitochondria, there was one mitochondria become two. 1, 2. Or in a diagrammatic way, we can see that it was 1 and then split in the middle and they become 2. And I also would like to thank for the resources that I have used. I hope you understand, students, the topic that we just discussed about the development of theory to explain the origin of cell. See you. Bye.